everybody. So I asked to come and talk a little bit about trends that we're seeing in cell therapy manufacturing. So before I dive in, I figured I should give you a quick introduction to where I come from so you know what my biases are here. Somewhat flippantly, I work for GE Healthcare Life Sciences. We're the part of GE that deals with anything that's wet and squishy. So we provide tools, technologies, and reagents to the research market, although our main focus is obviously on the manufacturing front, where we work quite closely with the pharmaceutical industries and biologics and antibody manufacture, as well as having a really strong focus on the cell and gene therapy market. All right. We are a global company. We have both research and development sites as well as commercial arms around the world. We're also very, very fortunate here in that we have great connectivity with most of the main manufacturers of these biologic and therapeutics. So we have the privilege to touch over 90% of all of the monoclonal antibody therapeutics that are on the market, and we currently touch all of the FDA-approved cell therapeutics. We also have the opportunity to come in not at just one part in the manufacturing workflow, but we offer everything from at the very front end, where we do hands-on services work, helping would-be manufacturers develop their processes and get those ready for manufacturing and scale up in a commercial environment. We obviously provide the tools and technology that we're very well known for, but we also go all the way now to providing GMP-qualified facilities for companies. So we really are trying to set ourselves up to be a full solution provider. And we think that puts us in a reasonable spot to talk a little bit about the trends that we're seeing. We ask ourselves, or try and ask ourselves, some critical questions through the process. Right? At the core, this is all about what can we do to bring therapies to market faster. But behind the scenes of that, it's all about how do we offer customers the flexibility and agility that they need out of a company, while still ensuring that they've got quality and efficient processes to drive for cost efficiency. The therapeutics that we're seeing are becoming increasingly complex, and the ways that you can manufacture them are becoming increasingly complex. So I think the question is, how do we do process optimization work in that milieu? And once we get the manufacturing process all worked out, we have to take a really hard look to say, is the supply chain and the logistical side of things that support that ready to receive that manufacturing? So see, these are some of the questions that we ask ourselves on a daily basis. So that leads me into trend number one. Fundamentally, this trend is really just about the field itself and the fact that the cell therapy field is moving incredibly quickly. We're up to over 1,000 clinical trials now in the space, which is almost 40% growth over just the last 4 years. And what's really exciting is that we're seeing a progression where these are no longer the academically driven phase 1-2 trials. We're starting to see almost 10% of them now hitting phase 3. And we're seeing therapies getting regulatory approval and coming out on market with a couple of really key cancer therapies that have launched in the last several years. So the field is maturing, but it's maturing at a tremendously accelerated rate relative than what we've historically seen for even the biologics field. So that means that that you're having to work with clients that are struggling to get to market as quickly as they can and are having to make some really critical manufacturing decisions on limited information. So that's something that all of us in the manufacturing space have to kind of realize and have in mind as we're trying to design strategies to assist them. The second trend is about the therapeutics themselves. These therapeutics are becoming increasingly complex. 30, 40, 50 years ago, drugs were fundamentally small molecules built from chemistry. And it's not to say that the manufacturing of those drugs was easy. It certainly wasn't by any stretch. However, fundamentally, those building blocks are just more simple in nature, and that means that the manufacturing approaches you have for them can be simpler and more similar to each other between products, typically. And even with the biologics and the monoclonal antibodies, the complexity is increased, but you're still coming off of a fundamental similar building block for most of these, which means you've got similarities that you can take advantage of when you're developing manufacturing processes. Now enters cell therapeutics, where you can go anywhere from, first off, you're starting with a human cell with all of the diversity that brings, and maybe you're just washing it and putting it back in to try and serve a purpose in another part of the body, all the way to the other side where you're going in and you're making fundamental genetic modifications to change the function of that cell. And that means that you're having to deal with a tremendous range of applications and indications, as well as ways that the cell product needs to be manufactured. So that complexity has profound implications in terms of how you need to develop your manufacturing, approaches, and how you ensure that you're having a safe and efficacious dose come out at the end of that. So this increase in complexity is a trend that we think we'll continue to see in the future as new therapeutics come on. If we dive into that a little bit and we look at the manufacturing of those therapeutics, you can loosely break these into the patient-specific approach, where one batch is one patient's worth of dosing, or the scale-up bulk product allogeneic approach, where you can bulk large quantities with multiple doses that then go out to see different patients. But if you break those down by the unit operations that are inherent in their manufacturing process, and you start to look for what are the problems that are inherent in here, in today's current milieu, you find actually that there are a number of similarities that come out that are driven around efficient manufacturing operations. And that makes a lot of sense if you pause for a moment and say, all right, well, where are we in the development life cycle? So 3-4 years ago, all of these products were fundamentally still in the research labs or the very early clinical trials, treating small numbers of patients. We're now progressing and we see the first commercial launches, and we're working hard to try and say, how do we get broad market acceptance and grow to clinical acceptance and global manufacturing scale? We know that for that to be successful, we need to change away from the scenario where we have been, where we're pulling in kind of whatever products and tools and technologies have been available for manufacturing of other therapies and kind of cobbling them together, which works all right if we've got 10, 20 doses per year. But as you move to thousands or potentially even tens of thousands of doses per year for some of these products in the near future, you need to have bespoke, specialized equipment that's going to come into play, and you need to try and figure out how do we drive improved potency of our drug products so that we can manage the dose sizes that we need to generate, and how do we try and move away from the largely open manual processing that's been used to make these drugs today. So that leads me to trend number 3, which is the first of the true manufacturing trends. This is by far the biggest problem that almost every client I interact with today is suffering. They're transferring processes that have historically come out of academic labs that are done by well-trained, highly skilled people in hoods with open processes that works fine at 10 doses, but is just not scalable when you move to a large mass market approach. You can't build factories that are big enough, and you can't hire sufficient people to actually get into the hoods and make those. Additionally, no matter how skilled all of you are in the room are, the natural human error rate means that if you're trying to manufacture 10,000 doses, you make too many mistakes in the processes and you end up losing material at a high scrap rate. And at the end of the day, that means you've got patients who are in need of a critical therapy that are not receiving it. So there's a big push to say, how can we switch to full closer to decrease contamination risk? And just as importantly, try and move to automated platforms, right? Obviously, there are cost benefits that we think will come out of that, but your fundamental driver here is reducing risk and enabling scalability of these. So this is the transition that all of the leading companies that are putting products on the market right now are facing. And it's something that as we look ahead to that next generation of therapeutic manufacturers, they're being smart and they're trying to say, when do I need to answer these questions, right? And the answer to that is really during your early clinical trials. You don't want to be faced with a scenario where you've just gotten a successful phase 2, you're ready to go to phase 3, and you realize that you're still hands-on in the hood with a team manufacturing your processes. So that sounds fantastic. You've convinced me, I've closed, I've automated my process. So what's the next trend we're seeing? Oh, sorry, I forgot the case study here. So this is an example of a CAR T process. Obviously generalized, but it comes from a number of the clients that we've worked with, where even these most advanced, you're talking 10 plus unit operations to carry out one of those things that are typically still in T-flasks or gas permeable bags for a whole thing being done in the hood. So this is how do you work with the companies that are out there, the GEs, the Sartoriuses, the Miltenys, to say, how can I take the manual process that I've developed and transition that into these systems? And it's much more complex than just taking something, a fight called gradient separation and saying, all right, well, I've got a machine here that runs a fight called separation. I've got a manual fight called separation. That should be an easy transfer. The details are that there will be product changes from that transfer. Maybe they're important, maybe they're not. So the underlying challenge is how do you identify those and then work through putting together these linked unit operations to ensure that the changes that you have introduced don't change the safety or efficacy of your product. And that's something that people need to build time in during the clinical process. All right, so now I can transfer to where I was going before. So, all right, we've closed, we've automated. Fundamentally, I've still said we've got upwards of 10 individual unit operations that are required to manufacture one of these therapeutics, each of which has numerous steps. So even if I've automated each individual process operation itself, I still have a tremendous number of transfers between processes, and I'm probably working with equipment from multiple manufacturers that doesn't talk to each other at all. So there's a big drive to say, how can I simplify my process? How can I move from 10 or 15 individual automated devices to 1, 2, 3 devices? And this introduces the fundamental approach of, all right, there are different philosophies here. Do I want to move to a one-box solution, something like a Miltenyi Prodigy, that has great advantages in that you can sit a single box, and after you've gone through the pain of transferring your process over, it's contained and it works great in small-scale manufacturing because you can have an operator come in and use that one device, and you can set that device up at another site at another global area and have a reason to believe that that's going to perform quite well. The downside to the single box approach is that as you scale up and you start to have factories producing thousands or tens of thousands, you probably have 70 or 80% of that one box that's sitting idle at any one time, because it's held up by one of those individual unit operations that is the rate limiting step. So there are other companies, GE falls into this bucket, where we say, all right, well, what about a modular approach, where we look for the natural synergies between your individual unit operation steps and the logical breakpoints, and then we try and put in a reduced number of unit operations so that as you scale up, you can purchase or design different numbers of those therapeutics into your process so that you're maximizing efficiency for each of those. Both great strategies, both pros and cons. Absolutely something that manufacturers need to be, or therapeutic manufacturers need to be thinking about, though, to try and make sure that they are building cost-effective and logistically friendly manufacturing processes. Okay, again, we're sold. We've closed, we've automated, we've simplified. Then you turn around and you pull out your perhaps multi-hundred-page paper-based batch record and try and walk into the clean room with it. It works, you can certainly manage that, but again, from a process and a logistical perspective, you know that there are advantages that can be taken by moving that to a fully electronic system. So this is the next trend we see, where the groups are trying to fully digitize their processes. Get all of their protocols electronic, get their batch records electronic, and take advantage of the fact that, okay, well, now I have a lot of automated systems, and those automated systems are continuously generating data for me. How do I start to pull that data in so that I have a better understanding of what this process looks like? That gives me strength in my regulatory and quality release standards, and it also helps me hopefully identify areas for improvement in my manufacturing process. Now, why people aren't doing this today is the fact that even though you've reduced and simplified, you still probably have half a dozen or more different unit operations or different devices from different manufacturers in your manufacturing group. Layered on top of that, you've got multiple software systems that are trying to manage the logistics, the supply chain, and the release of your products, and your quality management system. Getting all of those to feed in and talk to each other is quite complex. So this is where groups like Traxel, like GE, are trying to put together overarching programs that can suck in all of that information and start to output it in a format that's going to be useful for a manufacturer and simplify their logistics of manufacture here. One example of a program like this is the Chronicle software from GE, which effectively sits over a manufacturing suite, has each individual unit operation, whether it's a GE system or another system, providing the real-time output, providing opportunities for the users to come in and manually enter data that can't be transferred in an automatic method. And it gives full visibility. At the simplest level, you now have an electronic record, which can add a lot of faith and convenience when it comes to auditing, but it also lets you start to be forward-looking and say, all right, how do I preemptively say I've got an alarm in a system? And let someone who's sitting in a control room understand exactly what's happening and realize, who do I need to get in the room to fix that right now before I potentially lose the system or lose the therapeutic product? All right, so those are 5 key trends that are driving today's discussions with therapeutic manufacturers. I want to take 2 or 3 more minutes to quickly highlight 2 trends, which is where I see the field starting to go in the next 3, 5, 7 years. The first one is around process analytics. Right now, for the most part, groups are still manually taking samples and are being reactive in their approaches to how they use those. Where am I today? Okay, this is now the decision I need to take right now. Or even, where was I yesterday? Was that okay? Yes. Phew. No. Uh-oh. So process analytics is the concept of how do I take the information that the devices and manufacturing processes are already generating and start to move to real-time data collection. At a base level, that lets you make real-time decisions. And even more attractively, it starts to say, well, can I work with groups like Julie, who's got these great algorithms to get predictive? Can I look at what's happened on day 1, 2, and 3 of my 10-day manufacturing process and either predict whether I can harvest on day 8, 9, or 10, or realize that I'm starting to trend off track and perhaps there are things that I can do on day 4 to get me back on track so that I don't lose that sample. So incredibly powerful opportunities that I think is where the field is going to be moving. Frankly, this is things that are already commonplace in the in the biopharma biologics industry, and we just need to take advantage of that learning and figure out how to adapt them in the more complex biological systems that we're working with. So a really simple example of this comes from one of our pluripotent stem cell projects, where we're growing pluripotent stem cells in stirred tank reactors. Those reactors already have pH sensors. They don't typically have cell counts. But because of the fundamental relationship between how pH changes in a biological system, you can build corollary models that, based on changes in pH, allow you within a well-defined cell system to predict what your cell densities are. Now, instead of coming in and taking samples every day, we can start to make perfusion feeding decisions and harvest decisions purely based on real-time surrogate data sets. So this is a good example, I think, of where the field will start to go in the next couple of years. The other big one is cell engineering. And obviously, things like CAR-T have shown the power of going in and genetically modifying cells to change or improve their function. So that's going to continue, and I think we're going to see a new class of really strongly potent drugs that are being created around this. But behind the scenes, the other thing that this is doing is this is starting to build a comfort level with the regulatory bodies about the safety of genetic modification, as well as kind of the rules that can drive how that will happen. So from there, it's a relatively short putt to say, well, now what about starting to put safety switches into these new therapeutics, giving the physicians control to dial in how potent the drug is based on how any individual patient is responding. And from there, it's not much of a further step to say, can I make genetic modifications that make my manufacturing easier? Can I make these cells grow at higher densities? Can I remove the need for some of these really hard to source, qualify, and costly reagents so that they'll grow in a leaner condition? If you look again at the monoclonal antibody space, one of the biggest drivers of that has been the increase in titer from the cell lines. And the majority of that titer has been driven by improvement of the cell line, not the process optimization, which is where we as the cell therapy industry are all focusing our time to try and get those first steps. So I think more and more we're going to look at genetic modification as a powerful tool that lets us not just adjust the therapy itself, but improve the safety and manufacturing efficacy of those therapeutics. So, a couple of big trends that are in the field today, along with a few where we're going. Fundamentally, the takeaways are the field is moving quickly and the complexity is increasing. That complexity is going to require us to have automated, simplified, and digitized manufacturing processes if we want to be able to scale these to a true global offering. We know that analytics are important, and we're not using them to the full extent that we can. That's an advantage and an opportunity for us all to look at as we go forward. And genetic modification, I think, will be a continued and growing import in the next generation of therapeutics that are coming through. So, thank you very much for the time. Appreciate it.